Greetings, everyone. I'd like to try to answer in my own way the question which many of you are obviously curious about, and that is, how do cysts form? And this is not a clinical talk, so all the admonitions that Alexis gave about clinical advice won't be applicable here. And I should also add that this is my attempt as a biologist to explain cyst formation, but there are many publications in the literature that if you look at them, this is a seemingly simple question on its face, but a very complicated one to answer, and not everything is really known. So polycystic kidney disease is defined by the presence of numerous cysts in both kidneys. And these cysts originate from the tubules of the nephrons, which are the functional units of the kidney. And we'll be talking about that in a future webinar, how the kidney works, basically. And cyst growth involves two primary functions that under some circumstances are entirely normal and actually required, and under other circumstances are what we would call pathophysiological. In other words, they're inappropriate. So these two events are cell division, which is straightforward. Cells divide, and that's how organisms grow, and secretion. Anyone knows about secretion. Just looking at a meal, you'll be secreting from your salivary glands. So today I'll discuss the role of cell division and to a lesser extent secretion in the development of cysts in polycystic kidney disease. So we all start out as a zygote, and what you're looking at is an egg. That's the large circular object in the screen, and you're looking at a collection of sperm who are all vying to coalesce with the egg to form the zygote. And if you look at the sperm, you see at their head is the DNA that comes from the father, and within the much larger egg is the DNA that comes from the mother. And the DNA from the sperm and from the egg combine to form the zygote. So in PKD, either the egg contains the PKD mutation or the sperm does. So that's why, as an autosomal dominant disease, it's 50-50. So cell division, this is the basis of life. So this is the first division, and you're seeing an egg that has divided into two cells. In this type of division, it's actually not much larger than the original egg. The cytoplasm is shared, meaning the contents of the cell is shared between these two daughter cells. This little area here, and you'll see that on the right side as well, are called polar bodies, and they're of interest to researchers, but I'm not going to get into that today. So from the first division through to multiple divisions, you see that the cells are getting somewhat smaller, but they're more numerous. And this goes on until you have an entire human being. So mitosis, or cell division, is critical to life. So what does cell division accomplish? Well, very simply, it increases the number of cells, and it increases, and this is very important, the number of cell types, and this is called differentiation, because the egg, once it's fertilized, gives rise to every type of cell in the human body. The DNA contained with equal well, not quite equal contributions from mother and father, are all there and have the genetic information to make a human being out of that first cell. So growth in a multicellular organism requires cell division, and growth of individual cells, which sometimes happens. For example, neurons can be 4 feet long. This is called hypertrophy. And both growth and differentiation require cell division, and oddly enough, it requires cell death. That during embryogenesis, for example, the hand, which we all know has a thumb and 4 fingers, at some point looks like a paddle. So the tissue between the fingers and the thumb disappear by a controlled mechanism of cell death called apoptosis. The second P is silent, but you'll hear it pronounced apoptosis, even by people who ought to know better. So mitosis is an orderly process that results in the duplication of DNA and the condensation of that DNA into paired chromosomes. Now, by condensation, I'll show you some images of what that means, but normally your DNA exists in as sort of a scattered string, and condensation means that it sort of tightens up into what looks like chromosomes, and I'll show you what those chromosomes look like. And the result of this is two daughter cells that each have the same DNA of the parent cell. There are exceptions. For example, the cells in your bone marrow that make platelets often duplicate their DNA, but they don't actually divide, so that they have multiples of your DNA, and that's so that they can spin off platelets more efficiently. But that's another topic. Although the daughter cells contain identical DNA, they may be in fact the same as their parent cell. That's a symmetrical division, or they may be different. And at some point, that egg that I showed you and the two cells after the first division, which are basically pretty much identical daughters of the egg, eventually become differentiated. So you have the beginnings of cell types. So that's really what mitosis is all about. And technically, because of the platelet example I just gave you, mitosis is followed by something called cytokinesis, which is actually the physical division of the cell. Mitosis is more involved with the duplication of DNA and making sure that each of the daughter cells contain the same DNA. So I'm hungry, it's lunchtime, and this is sort of an example of what happens. So you have in the upper left-hand Bismarck icing that shows the nucleus in that cell. And in early anaphase, and these terms aren't important unless you're interested in this sort of thing, begin to show some condensation of the DNA in the nucleus. You see in this first Bismarck, this is called an interphase cell. In other words, it's between mitosis, or it may be post-mitotic, never to divide again. But if it is going to divide, you see this condensation of what's called chromatin, which contains the DNA. And at this point, the cell has synthesized a duplicate copy of all the DNA and all the chromosomes. And in metaphase, you see that there are two poles now on the Bismarck. This is the upper right. And you can see these strands that are called microtubules. And this is the metaphase plate that contains the chromosomes, the pairs of chromosomes that are going to be pulled apart. And this is early telophase, which is on the bottom left. And you can see that the microtubules are getting smaller and the chromosomes are being pulled apart. And in late telophase, you can see the beginnings of two separate cells, although they haven't actually gone through cytokinesis yet. And you can see that the chromosomes are beginning to unravel again to resemble more the interphase nucleus in the upper left. And then finally, in a miracle of biology, you have two Bismarcks with identical nuclei and identical DNA. And in this case, you'd call it a symmetrical division. In other words, you've got two daughter cells that look just like the parent cell. In reality, and it's often when you take a biology class, you look at plants because A, their cells are beautiful, B, they come in nice square shapes because they have chitin, not chitin, but cellulose that defines the borders of the cell. And if you go from left to right in the upper panel, you see an interphase nucleus the very beginnings of condensation in the next panel, more condensation in the middle upper panel, and you begin to see the formation of chromosomes in late anaphase and more identification of individual chromosomes in early metaphase. So if you go to the bottom panel, you can see what's called the metaphase plate, and these are all the chromosomes. They're all paired. You can't see them as pairs very easily here, but if you go to the next panel on the lower left, you can see, assuming you see my pointer, that this is a duplicate of this chromosome, and that these are all paired up. The most obvious one is sort of middle right, and you can see sort of the V-shaped chromosome that's going to go to the top of the slide, and the U-shaped chromosome that's going to go to the bottom of the slide. And the next panel shows exactly that. Here are all the chromosomes that are going to be part of a new cell, and here are the chromosomes identical to those that are also going to be part of a new cell. And so you see them separating further, and finally you actually see cytokinesis. You can see this division, and now you have two cells where you had only one. So from the upper left to the lower right, you can see basically the entire sequence of cell division, mitosis. In some other reality, because in looking up these images on the web, this was included in images of mitosis, and I was just stunned by how beautiful it is. And this has more relevance towards a certain area of mathematics, but this is really what's going on as cells separate. This would be telophase, where you can see these two daughter cells who are still sharing some cytoplasm together, about to be pulled apart. And I just want to convey to you the elegance of what goes on, and this is really one of the fundamental features of multicellular life. So what about cell division in daily life? Well, it's to replace exhausted, dying, and dead cells, and that probably is obvious to all of you. Your skin at the surface is dead, and you ultimately lose those cells, and they're constantly replaced with new ones by mitosis and differentiation. And so the skin is a very obvious example, but the lining of the gut is replaced, essentially, the epithelial lining of the gut, in a matter of days in some cases. So those cells, as they get older, they go to the tip of the villi in the intestine, and they're desquamated. Again, a big term, but it means they end up being shoved out of the body and replaced by new cells. And of course, if you've ever, as everybody has, cut yourself or suffered a minor injury or even a major injury, you know that the body has capacity to heal. So fibroblasts come in and they divide and they form scar tissue, and in some organs like the liver can reconstitute itself if a lobe is taken surgically, for example. And another area where there's a lot of cell division and cell differentiation is the bone marrow, and this replaces tired red blood cells. Our blood cells have a certain half-life, meaning after about 8 to 22 days, I forget what the actual half-life is for red blood cells, they're taken up by the spleen to be broken down into their component proteins like hemoglobin and recycled. And the bone marrow produces new red blood cells as required. And part of the reason I use this as an example is that the kidney makes erythropoietin, which is a hormone that tells the bone marrow when to make red blood cells. And for those who have anemia with chronic kidney disease, they often need injections of erythropoietin to tell the bone marrow to be active in making red blood cells. And the bone marrow also contains the precursor cells to white blood cells, and these can multiply rapidly in response to an infection. So cell division is a real part of our daily life. But certain cell types, like in the heart and the brain, under most circumstances are post-mitotic, and they live for a long time. So cell division in daily life, for some of us, unfortunately, cell division is inappropriate. And hyperplasia, again, fancy term, but really it just means too many cells. This occurs actually in response to injury, so it's not really a pathology. It's what's required to cover the defect in your skin, for example, if you've fallen off your bike and you have a scrape. Dysplasia means cell division, but that the cells are different from their parent cells. And under normal circumstances, they would be the same as parent cells. And this can progress into neoplasia, which very simply means new growth, but it's dysplasia without respect for boundaries. In other words, and this will come into my explanation of how cysts form, because I realize I haven't spoken about that for quite a while, that cells generally respect their boundaries. In other words, if they divide and come up against an adjacent cell, they're not going to divide again because that adjacent cell is telling it, we don't need more cells in this particular location. But in cancer, generally, these dysplastic cells lose their respect for boundaries and they head out into the bloodstream and populate other tissues. So now we get to cyst formation in the kidneys, whether it be autosomal dominant or autosomal recessive. And what it is inappropriate cell division in tubules of the kidney, or the nephrons of the kidney. And so what is the fate of these daughter cells of the inappropriate division? That's really the question. Now, the kidney does have cell division. It suffers injuries, particularly if you had a small stone, for example, and some of you may have heard the webinar last night about stone formation. Many of these stones form just for a second or two, but they may scrape a cell in the kidney, and that cell has to be replaced, so its neighbor divides and replaces that injured cell. So in PKD, the first hit, so to speak, is the mutation in 85% of the cases, PKD1, and in 15% of the cases in PKD2, these are genes that define two proteins involved in ADPKD, and those that have the PKD mutation, as many of you probably know, have a much milder course of the disease, but these two proteins are required to interact with each other appropriately, and if they don't, that's what leads to cyst formation. So that first hit is the actual mutation, and each family tends to have a fairly unique mutation that's probably shared by only 1 or 2% of other people with PKD. So then there's a second hit, and that is, why does the cell with this mutation respond to a stimulus by dividing? The normal cells don't. And what's also another very interesting question is that all of the cells contain this same mutation, but only a few of them actually divide. So something else has to be going on, and this is what I've called other hits. And that may be that, remember I told you that the egg contains the maternal chromosomes and the sperm contains the paternal chromosomes, and one of those chromosomes contains the mutation. So that means that one of the copies of DNA has a normal PKD gene and another has an abnormal PKD gene with the mutation. So some people think, and I think there's evidence for, something called a somatic mutation, where the second copy of the PKD gene that comes from either the mother or father that was normal has an inactivating mutation such that there is no source anymore for the normal product of the PKD1 gene, which is polycystin 1. So that's almost for another talk, but in any case, you get two daughter cells which each then divide again, and this results in 4 cells, and the 4 daughter cells each divide, and so on and so forth, 2, 4, 8, 16, 32, 64. These are actually the powers of 2. 2 to the second is 4, 2 to the fourth is 8, and so forth. And 2 to the n is where you really get into trouble with PKD, because that means these cells have been dividing for a long time, and that the consequence of the powers of 2 can be enormous, both literally and figuratively. 2 to the power of 60, for example, is a very, very long number, and it also implies a very large number of cells. So this is my conception. I put this together myself, and if you'll bear with me, this is how I visualize what's happening in a renal tubule. And you can see that these are cuboidal blue cells, and here's the renal tubule lumen. And for whatever reason, for whatever signal, and that's where the question mark is, a cell with the PKD mutation divides. And what happens? There's no room for that division because it was inappropriate. So these cells are no longer in an appropriate position in the epithelium of the tubule. So there's a little bump. At this point, even a pathologist looking at microscopic slides probably wouldn't notice this. But if these cells continued to divide, and again, so there were 4 cells. Now, if you look at the arrow, if each of these cells divide, you have 8 cells. And because the tubule still continues to exist as this tubular structure, these dividing cells have nowhere else to go but to sort of bleb out. And this is really the beginning of the cyst. And this process continues for some time. I'm showing you, I think, 4, 8, this is after multiple rounds of division, that you now have an obvious cyst. The tubular fluid is going through what remains of the normal tubule, which is most of it, and there's still some continuity between the cyst and the urine flow through that tubule. But these abnormal cells are also secreting into this bleb, which you can now call a cyst, and that's contributing to its growth and size. So now we're at the 4th slide in this sequence, and this is 2 to the 5th cells. So I think this is 32 if I counted correctly in creating this, and it is, if you notice, lost all continuity with the tubule below it. So here are the 2 original daughter cells, and now there's this blind cyst which continues to secrete fluid into its lumen, which is the space that the cyst occupies. Just think of it as a balloon that's had the knot tied, and it continues to grow and divide. And these cysts become larger, they become big enough to see with the naked eye, and they attract an inflammatory response, and they crush adjacent normal tubules. This tubule could be one of them, where this layer of cells gets crushed into this layer of cells, and urine can no longer form. And that's why this cyst formation is ultimately so injurious to the kidney. So these cysts form, and they may form very early in life without your knowledge. In other words, you feel just fine, your renal function is just fine, but they inexorably grow over time, and it's been determined that that's about close to 6% per year compounded annually. So these cysts are continually forming and growing, and that's what leads to something called total kidney volume, which is a very valuable biomarker that the FDA has just issued a draft guidance for, and I'd like to thank all of the scientists and the foundation for the 8 years it took to qualify total kidney volume as an enrichment biomarker for clinical trials, that total kidney volume is involved in this 5.7% growth. Maybe it's a little lower in those with PKD2 mutations, or maybe it's just that they have fewer cysts. So the questions that, as a patient, I know I asked is, if all my cells have this same mutation, why do only some renal tubule cells respond in this way, meaning that they divide in a sort of uncontrolled fashion and they secrete fluid derived from the blood, and why only some? You know, it may be only 1 in 500 to 1 in 1,000 nephrons where you actually get this. So that's one question. And I think that's what the second hit may be. It may be that a cell is injured and there's inappropriate cell division that continues rather than stopping. Why do they keep dividing? Well, it may be because of the secretion, that the physical pressure of the fluid in the cysts, and this I'm strictly hypothesizing on my own now, may be a signal for division. But many of the drugs that are being looked at for treatment of PKD are anti-proliferative agents, which are similar to the kind of drugs that are used to treat cancer. But obviously for PKD, we want anti-proliferative drugs that only affect cells in the kidney, not all over the body. So a lot of research, and the foundation is funding it, is looking at the second hit. There are questions as to whether there's a third hit, and are secretion and mitosis related? I've given you my opinion that I think that they are. And I'm happy to tell you that the progress that's being made right now in addressing this fundamental question of why do cysts form and what can you do to stop that growth is being actively investigated by scientists and clinicians, thanks in part to your contributions and the ability of the foundation to fund an increasing number of fellowships and basic research grants that will get to some of the complicated answers that are involved in answering these questions. For those of you that heard me talk about cilia, I didn't mention them at all, but that's still another core interest in why polycystic kidney disease develops as it does. So in conclusion, cyst formation and growth are inexorable facts in PKD. This is really what causes the disease. And behind that simple sentence is an incredible amount of research, especially when you look to what was known about the disease in 1982 when the foundation was formed. And there are multiple therapies that are now being designed to slow the growth of cysts to preserve normal kidney tissue. In other words, if the cysts grow at a much smaller rate, they won't impinge on the remaining normal kidney and cause inflammation and high blood pressure and loss of those normal kidney tubules. And there are many targets. Some of them are pointed directly at the mutated protein, the mutated polycystin 1 or polycystin 2, and many of them are involved in what we call downstream events that are somehow the result of those mutations, and that's this inappropriate cell division and secretion into cysts. Recently in the literature there have been many exciting concepts that I hope will have clinical application, in that you have an investigator named Dr. Kelly at Indiana University has shown in rodents at least that she can inject normal renal cells into a PKD model rodent, and these PKD affected rodents, with the injection of these normal cells, actually show some correction of the defects reflected by the polycystin proteins. This is very exciting because although whole organs are typically rejected by the body, and that's why you need anti-rejection medications, in this case, and this may be just rodents. They're remarkably adaptable, but they don't recognize individually injected cells as foreign. So at least in this early model, and I'm not saying this will be in the clinic next year or in the next 5 years, but it shows you that I think there are enough approaches to correcting the basic defect in PKD that we are going to see not only something that slows growth, but therapies that may actually stop it. So I'm more than happy to take questions now. And I just wanted to let you know of upcoming education events. I'll be talking next month on September 16th on basic kidney function or renal physiology 101. And there will be evening webinars on October 14th, the emotional toll of PKD. I could probably talk about that as an individual, but you won't be hearing from me. And then on December 2nd, you will hear from me again, and I will try to relate to you my experience in big pharma and tell you what are the hurdles and issues involved in bringing a new compound to the market. And again, reminding everybody of the PKD National Convention in Orlando, Florida next June, and stay tuned to pkdcure.org for updates. So with that, I'll take questions.
Okay, we have several coming in. One, and this first question is a little more clinical in nature, and so I will reiterate that David is a basic scientist, not a physician, but I think he probably can apply some of his robust knowledge to helping to answer this question. So they're wondering the impact of caffeine on PKD, and I don't know if there are studies that imply that caffeine stimulates growth, or if that's kind of a rumor. I don't know. Are there studies that show that? And if there are or not, what's your opinion?
So one thing I did when I was in industry is preclinical toxicology. In other words, by mandate, the FDA requires us to study rodents and in vitro cell systems for toxicity. And caffeine, the reason that you've heard that it might not be good for PKD is that it can stimulate cyclic AMP. And in PKD, activation of cyclic AMP also activates cell division. However, as Paracelsus once said, and this was a long time ago in the 17th century, it's all a matter of dose. So I would talk to your nephrologist about whether he or she thinks it's advisable to drink coffee or not. Coffee has many benefits, but I have the feeling they might tell you 1 cup is not an issue, but if you're drinking 6 or 8 cups, it may well be an issue, even if you didn't have PKD. And obviously the colas contain caffeine. Chocolate contains a little caffeine. So, well, it's another form of a molecule like caffeine. So, yes, there's some substance to your question, but you need to ask your nephrologist.
Perfect. So this is an interesting question. Do people without PKD develop cysts in a different way, or does your explanation of cyst formation hold for other cystic issues?
That's an extremely good question, because sometimes during an ultrasound, which is diagnostic for PKD if you're 30 or over, and you see cysts bilaterally, in other words, in both kidneys, sometimes you will have an ultrasound done and you do not have PKD, but you will see a cyst or two in one kidney. So as to whether those cysts are formed like they are in PKD is a good question, and I don't honestly know the answer. But cysts that are diagnosed by MRI or ultrasound, you don't know exactly what their origin is. Sometimes their origin is in the glomeruli, or they're formed as a result of blockage of a nephron, and it really just blows up like a balloon, but doesn't really involve cell division and secretion in the same way that it occurs in PKD. And very often the presence of one or two cysts, particularly as you get older, is a coincidental finding that doesn't have much physiological significance.
So, follow up to that, can you comment on if you have PKD and you have liver cysts or PLD, do the cysts form in the same manner in the liver as they do in the kidney?
So in the liver, the bile ducts are where the cyst formation occurs. These are obviously different types of epithelial cells than in the kidney.
And what's an epithelial cell?
The epithelial cells are what lines the tubules or the nephron of the kidney, and that's what we've really been talking about all this time, that the cells doing the division and forming the cysts are epithelial cells gone wrong, so to speak. And the same thing does happen in the kidney, and it starts in the bile ducts, the tiny ones, not your big bile duct that drains into your intestine. But the liver is a fundamentally different tissue than the kidney. So these cysts are significant more for their physical size than for their effect on liver function. You can have many cysts and you can have a liver that is so large that you may need a transplant, but your liver function may still be normal. So unlike the kidney, where the cysts really have a profound effect over time on kidney function, cysts in the liver rarely do.
Okay, thank you. Okay, we have got several questions here. So one person, I think, got a little bit, several people looking at the questions got a little bit lost in your explanation of PKD1 and PKD2. So although this could be like a topic for a whole new webinar, maybe we should consider doing that. Could you do a quick run through with that again? And then someone else is kind of saying that they've heard that people have had a negative result for the PKD gene, but they still have PKD. So is it possible, or basically can you explain PKD1 and PKD2, the gene, the mutation, how all that works?
Yeah, well, Alexis is right. That's a talk in and of itself.
And we were looking for a topic for November, so maybe we've just found it.
So polycystin 1 is an incredibly large protein that is the instructions for polycystin 1, if you will, is in the PKD1 gene. And polycystin 2 is a much smaller protein that is the product of the PKD2 gene. Now, these two proteins, polycystin 1 and 2, interact, and they interact in the cilium. And some people think that this interaction leads to signaling. In other words, that the cilia is bent by the flowing urine, and somehow the interaction between these two proteins, polycystin 1 and 2, leads to a signal, which might be calcium moving across the cell membrane. It might be something else. This is a topic of active research. But mutations in the much bigger polycystin 1 tend to lead to the more typical disease where end-stage renal failure can occur in the 5th or 6th decades of life, whereas mutations in the much smaller protein, polycystin 2, generally lead to a much milder phenotype, meaning that the consequence of that mutation is milder, and these patients typically may not end up in end-stage renal failure. But I can talk about these proteins more. They're very interesting and complicated proteins. And to the second question about genetics, The mutations that occur in polycystic kidney disease are not simple base pair mutations. In other words, in DNA, if you've seen the DNA ladder as discovered by Watson and Crick, there are base pairs, and some mutations are a single base pair. But in particularly polycystin 1, there can be truncation mutations where large parts of the protein are never even expressed, and you get a remnant protein or no protein at all. So the genetic tests don't necessarily have sensitivity for the entire variability of the mutations that define autosomal dominant PKD. So there is the possibility for false negatives in genetics.
Okay, very good. We're getting some good suggestions for different things that we can consider, possibly adding some resources, web resources and things that people can read up on some of the stuff after the fact. So we'll look into that for future ones, obviously not for this one. Let's see. Let's see. So we're having some questions about blood pressure, cyst growth, cyst formation. There's some specific questions, but maybe could you just comment generally on blood pressure, cyst formation and growth, how they interact, and then if there's some specific things in here that you don't address, then we can follow up with those.
And again, these are questions that the clinical researchers in PKD can much better answer. So don't take my answer with a grain of salt, however, because that's not good for high blood pressure. But what happens is that, in my understanding, this is a physical issue. That just imagine two balloons that are blowing up, and there are normal kidney tubules that are caught between these two enlarging balloons. And what happens is they tend to compress the arterioles, which are tiny little arteries that supply the kidneys with blood. And when that blood supply gets constricted by these growing balloons, the kidney sends out a hormone that tells the body to raise blood pressure. You know, the kidneys being relatively starved of blood, so it sends out a signal, and your blood pressure goes up, and it may help that particular part of the kidney, but you also suffer the consequences of high blood pressure in general, involving arteries having nothing to do with the kidney, such as in your heart or in your legs. So it's really the physical compression of the blood vessels and the normal nephrons that are thought to lead to hypertension, which is one of the most common early symptoms of the disease.
Okay, so as a follow-up, someone is asking about the HALT study, which I know you're familiar with, David. And they're saying basically we know that it showed that keeping blood pressure, the idea was that keeping blood pressure low helps slow the progression of cyst formation. And I'm not sure that that is entirely accurate. That's not necessarily what was discovered. Am I correct?
Correct.
So maybe you can kind of comment on that, and then they're just wondering, because it's not necessarily easy to overcorrect blood pressure because of side effects of medication, so can you kind of just comment on the HALT findings and its impact? Well,
the HALT studies looked at two drugs, the ACE inhibitors, which stands for angiotensin converting enzyme. This is an enzyme that takes angiotensinogen and makes it into angiotensin. Again, please excuse the large words, but if you just think of angiotensin, think of tensin as tense. So angiotensin converting enzyme contributes to high blood pressure because it releases angiotensin. Angiotensin works by coupling up with receptors that cause your blood vessels to react and tighten to increase pressure. It's just like with a hose, that if you put a tight nozzle on it, you'll get more pressure of the water coming out. And they also looked at a drug called an angiotensin receptor blocker. So these are two drugs that get at the same mechanism, basically. And Halt looked at whether one or the other were equivalent, or if you combine both, did you get a better effect? But I think the more important aspect of the study is whether to control blood pressure to something like 130/80 is sort of high normal, or do you want to correct it to a much more stringent, like 110/70 limit, which athletes have, for example. And the study, as designed, did not quite meet the predefined statistical significance, but it did seem to show that better control of blood pressure leads to better control of the disease, because hypertension has effects all over the body, not just the kidney. So it was an important aspect in that sense, and also it was and is the biggest PKD study that I'm aware of to date.
Great. So a lot of good data gathered for the HALT study, and that's HALT, H-A-L-T. Someone was asking for that, and I, for a long time, thought it stood, H-A-L-T was an acronym for something, and I could never figure out what it was. I finally asked one of the principal investigators, and they said, nope, it's just meaning to halt PKD. And I was like, you could have just lowercase because it's all uppercase.
There's a little more story to that. Many clinical trials, when you look, they seem to have, neat names.
They all do.
And what you do is you look at the titles to these clinical trials, and they're probably 3 lines long. And what the investigators do is they selectively capitalize letters until they find something that sounds good. So HALT may be found in the title to that entire protocol that was submitted to the NIH. So
then how do they get reprised?
You
don't have to answer that.
Yeah, that's for Otska to...
That's for them to know and keep close.
Yeah.
Okay, we do have a few questions that are unrelated to the topic, although very good questions. So we'll get to a couple of those here in just a minute, but there's one about cysts and tolvaptan. So can you talk about kind of what tolvaptan does? Does it affect cysts? How does it do that?
Well, the tolvaptan studies, to me, were very consequential, and it's led to the approval of tolvaptan, also known as Ginarc, in Canada and the European Union countries, and Japan, of course, but not in the U.S.
And the most recent study is the reprise study.
Yes.
Just to clarify.
And that was, I think, by request of the FDA for additional information. So what the study showed is that tolvaptan almost halved the growth of cysts. So I told you in the beginning of my talk that cyst growth is about 5.7% per year compounded. In the now I'm forgetting the name of the original Tempo trial, cyst growth was cut in half. So I think it had a very significant effect, but the problem with PKD studies is that the course of the disease for most people is decades, and clinical trials are typically for a year or 2 years at the most. So I think that tolvaptan did have a significant effect on cyst growth. It cut that growth by half, but many of the patients, because of our considerable renal reserve, in other words, obviously living donors can do just fine on one kidney, in the early course of PKD, even though you're losing normal kidney tissue over time, your function remains relatively normal until maybe the 4th or 5th decade of life. So the TEMPO trial, while showing a dramatic, in my opinion, structural effect, didn't really show as great a functional effect, but I think that's because many of the patients had essentially normal function to begin with, so it's hard to show a difference. Those are strictly opinions of my own, and you'll find many nephrologists with different interpretations of the study. So in no way am I either endorsing or not endorsing tolvaptan, but what I do know is that it did affect cyst growth.
If you are interested in learning more about the REPRISE study, you can go to pkdstudy.com, or you can check out your latest issue of the e-news, which I believe is coming out today, and there's a little bit of information there as well. And that is still openly recruiting for participants. The PKD Foundation does not encourage or discourage. We simply provide information about studies. You can also go to our website to learn more about the REPRISE and other studies available that could be available in your area, and that is on our website, pkdcure.org, and then you go to, if you can put clinical studies into the search box and you'll see that pop up. I don't remember the exact URL. Okay. There's a couple of questions here that are not really related to cysts, but we have about 5 minutes, and so we have several questions still outstanding that we're not going to get to, but I think there's one here that I can answer very quickly, and it's someone asking about a specific diet plan for people with PKD. Is there such a thing? And David, correct me if I'm wrong, but to my knowledge, there's not necessarily a quote-unquote PKD diet that has been studied and necessarily approved for recommendation. However, we do know that generally speaking, a healthy diet is preferable. High in water, so you want to drink a lot of water, and lower, I should say, in protein and high in fruits and vegetables.
Yeah, so there are dietitians, many of them, that specialize in the renal diet because it is a challenge to lower your protein intake, yet get quality protein that's required. Hydration, I think, and I've said this before, in general, is an issue for everyone, whether they have PKD or not. It is better to be hydrated than, by the time your mouth is dry, your brain is telling your kidneys to work harder to save water and concentrate your urine. And one of the issues with PKD is that concentrating ability of the kidney is lost over time. So you don't want to stress yourself by being dehydrated. And I would say, and I don't know the outcome of these studies yet, water intake and formulas for exactly how much water is appropriate and how much is too much. And again, I mentioned earlier that it's all a matter of dose. People running marathons have died of over water intake, such that they dilute the salt in their blood. So everything has its toxic limits, even water. So I don't want anybody to construe anything I've said as carrying around a gallon jug of water. And remember, when you eat fruits and vegetables, you get a lot of water from those types of healthy foods.
You'd have to work pretty hard to overhydrate as a PKD patient, though, don't you think?
Well, since your kidney, yeah.
You have to work hard at it.
You'd be excusing yourself rather often.
So I think we have one final question. We have 2 minutes to 1 p.m. And this question, I think, is something that we should actually probably consider doing even an evening webinar on. We get this pretty often. So someone is wondering, if you have a transplant, do you still have PKD? And can you give us the 30-second response, David?
Well, as somebody who has PKD and is nearing my 6th year with a transplant, thanks to a lovely person I worked with a long time ago, yes, you still have PKD. PKD, as we were just talking about, affects the liver. For some people, it affects blood vessels, particularly in the brain. You can often have cysts in other organs than the kidney or liver, and cysts have been seen in the thyroid gland, in the pancreas. Diverticulosis is more common in PKD, and those things are not cured by a transplant. So while a transplant has been night and day for me, and that's why I'm at this job, I don't think it would have been otherwise. You still have to be careful about some of the other issues involved with PKD.
Absolutely. Okay, I think that is all the time we have. We did leave a handful of questions outstanding just because I think that they would take more time than we have to answer right now, and so we will see what we can do about getting those answered, and then I have promised someone that we will work on getting all of the questions that were asked answered on paper, and we can post that along with the video. Someone requested that, and I think that's a pretty reasonable thing to ask. So we'll work on that. It might take us a little bit of time, but we'll get that posted as well. So thank you, David, for all of your knowledge. Thank you to the audience for listening, tuning in at this noon hour or whatever time it is in your time zone. You came at us with some really great questions. We had some good conversation, and we just appreciate the opportunity to provide you with the best possible education we can. Let's see, I'm going to email a quick survey to anyone who registered and attended, so look for that. If you have ideas for future topics, you can include that in a survey. You can email it to me at education@pkdcure.org or tweet it to us. Pretty much any way you can think to communicate with us, you can let us know that way. And I think that's it. On behalf of everyone at the PKD Foundation, thanks so much for joining, and we hope to see you next month.